We're going to take a look at a biochemical process that occurs in photosynthetic organisms such as plants and algae or photosynthetic bacteria called photosynthesis. So what we need to look at in terms of photosynthesis across this lesson is why do we have it? What is it used for? We need to have a look at where photosynthesis takes place, the chloroplast, and have a bit of a look at the structure of the chloroplast. Also going to talk a little bit about the bacterial origins of the chloroplast. Importantly, we need to look at the inputs and the outputs of each stage of photosynthesis. And then finally, some different factors that affect the rate in which photosynthesis takes place. So on this particular slide, to start us off, it says write a word equation and a balanced chemical equation for photosynthesis. And on the next slide, you can see an example of what yours should look like. But the equation for photosynthesis is important because if you can remember the equation, well, firstly, it could be a question in itself, just write the balanced chemical equation. But it also gives you a nice little prompt when you're trying to identify inputs and outputs, which are pretty common questions as well. So I would say for sure you need to memorise that photosynthesis equation. So what's the purpose of photosynthesis? Basically, it's a process that converts light energy from the sun into stored chemical energy in the plant in the form of glucose. So it's a process where we take carbon dioxide, we take water, and in the presence of sunlight and in the location of the chloroplast, a series of chemical reactions are able to take place that produce oxygen and produce glucose. Here's our equation for photosynthesis. So that one I said we need to learn off by heart. And as you can see, we take 6 carbon dioxide molecules and 6 water molecules as our inputs. And in the presence of light, sunlight, and chlorophyll, which is a pigment found in the chloroplast, we're able to produce 1 molecule of glucose and 6 molecules of oxygen. Just make a note when you're writing out this equation, you always need to include light above the arrow and chlorophyll below your arrow, because in the absence of those things, the process won't take place. And the reason we put them above and below the arrow is because they're not inputs and they're not outputs. They're not being changed in the process or converted into any other form. They're staying as is. But if we were to put them on the left-hand side of the arrow, it's implying that something's happening and changing to them to convert them into a different form as a product. So that's why we keep them above and below our arrow. Why does a leaf look green? Well, it looks green because it's got a pigment inside it, which is called chlorophyll. So inside our photosynthetic cells, which are the cells of the plants generally found in their leaves, those cells have organelles called chloroplasts, which I know you would be familiar with already. Inside the chloroplast, there's a lot of this small pigment called chlorophyll. And chlorophyll is a pigment that is able to trap light. So when sunlight shines down on a plant, that sunlight will enter the leaf of the plant, it'll enter the cells of the leaf, it'll enter the chloroplast in the cells of the leaf, and chlorophyll will absorb certain wavelengths of light. So particularly blue and red are wavelengths that chlorophyll is really good at absorbing. And by absorbing the light, it's trapping the light, and then it's able to use that light in the process of photosynthesis. However, whilst chlorophyll is good at trapping blue and red wavelengths of light, it's not good at all at trapping green wavelengths of light. And it's like a mirror to green light. As the green light shines on the chlorophyll, it's reflected straight back out of the plant, out of the leaf, and into your eye. So the colour you see is green because that's the only colour getting reflected into your eye. whereas the other colours are actually getting absorbed inside the plant itself. Chlorophyll, if we didn't have chlorophyll in a chloroplast, photosynthesis would not take place because there'd be nothing in there to trap the sunlight to use it for a particular purpose. Chlorophyll is not our only pigment. There's other pigments inside chloroplasts as well. We call them accessory pigments, and we'll have a look at them later. And they help chlorophyll. Chlorophyll is the main pigment that we use for photosynthesis. But the other ones are able to trap some of those other wavelengths of light so that we can make the process as effective as we can with all different wavelengths. Let's take a look inside a leaf. So let's go to a plant and let's go from the leaf of the plant inside an individual cell of the plant, and then this picture up on the right here is an image of a plant cell. In the plant cell you can see we've got all of our usual organelles that we're already familiar with, and then we've got this particular organelle, the green one, which is called our chloroplast. And if we take a look at our chloroplast, you'll see that it itself has some specific structures. So it's got a double membrane outside the chloroplast, which is enclosing the chloroplast. Inside the chloroplast, it's got all of these green little stacks, which look like little stacks of poker chips. And then surrounding all these dark green stacks is just a fluid-filled space that sort of bathes the stacks. We can't just call them stacks, unfortunately. We need to give them some biological names. So each individual disc is called a thylakoid. And a thylakoid stacks on top of other thylakoids to form a stack of thylakoids, which we call a granum. So a thylakoid is an individual disc. Many of those individual thylakoids stacked on top of each other forms a stack of thylakoids called a granum. And as you can see, there's lots of different granum in the chloroplast. So we use the word grana as a plural to talk about all of the granum stacks within the chloroplast. That fluid-filled space I talked about inside the chloroplast that's not the granum is called the stroma. And as we look at photosynthesis, which has two main stages, a stage called the light-dependent stage and then the light-independent stage. The light-dependent stage takes place in the thylakoids, in their membranes, whereas the light-independent stage takes place in the stroma. Some info over on the right-hand side here for you, not stuff you would need to memorise, but just some interesting facts about chloroplasts in terms of their abundance and their size. Read down the bottom here, it says the stroma of chloroplasts contains ribosomes and it contains DNA. And we should hopefully know by now that the nucleus is not the only part of a cell where DNA is found. We also have DNA found in chloroplasts, and we also have DNA found in mitochondria. And the DNA in a chloroplast or a mitochondria, because it's not housed within a nucleus, we call it naked DNA. And you'll also notice here that the chloroplast has its own ribosomes. So the chloroplast is actually really similar in size and structure to a degree to a bacteria. So it is believed that chloroplasts and mitochondria, way back in our evolutionary line, were actual bacterium. So a chloroplast was believed to have been a photosynthetic bacterium at some stage. And through a theory we call endosymbiosis, it's been engulfed by a larger bacteria, and over time that particular cell has evolved to develop other organelles, specialised locations within it. Something that indicates that chloroplast probably was a bacterium at some stage is its size, because it's basically the same size as an average bacterium, but also the fact that it does have DNA and ribosomes, which none of the other organelles do besides the mitochondria, which suggests it's self-sufficient. If it's got DNA and ribosomes, it can make its own proteins, and it does. And the proteins it makes and the genes on its DNA are all to do with the proteins required in the process of photosynthesis. Just a couple of questions for you to think about now, so you could pause this and have a go at answering them, and then I'll continue on with the answers. So, what type of reaction is photosynthesis? It's an anabolic reaction, if you link it back to our enzymes lesson, because it's building something larger and it's requiring an input of energy. So it's anabolic. What category of living organisms are plants? Plants are autotrophs because they're capable of sustaining themselves, really, sort of making their own food or their own nutrients. Describe the energy conversion taking place during photosynthesis. We are converting light energy from the sun into stored chemical energy in the form of glucose. So let's have a look at the actual process. So how exactly does a plant take in sunlight and use that sunlight to fuel a series of reactions that produces glucose and oxygen? So within the chloroplast, as I said before, we've got two specific stages. The light-dependent reactions, or the light-dependent stage of photosynthesis. It occurs first, and it occurs in the thylakoid membranes, or in the grana. And then following that, we've got the light-dependent stage, which is occurring in the stroma. Now, I'm going to talk through the process, but if you were to also look at the video on this YouTube channel called Photosynthesis, it'll take you through a detailed diagrammatical and annotated overview of the specific process. So I would recommend watching that one most definitely. So as we said, in our chloroplasts, we have chlorophyll, that pigment molecule that traps light. And the chlorophyll is specifically found in the thylakoid membranes. And what happens is when light enters the plant and it reaches the chlorophyll in the thylakoid membranes, it gets trapped by the chlorophyll and it causes the chlorophyll to get excited, if you like. And that excited state initiates a series of reactions that enables the light-dependent stage to form ATP and also to form some hydrogen ions, and it also releases oxygen as well. So once we block out that light independent stage on the other side, this is what we're looking at. So we're looking at the first stage of photosynthesis, the light dependent stage, and we've got a picture of our chloroplast up here, and we're just showing that the light dependent stage occurs here in the thylakoid membranes in the stack of grana, and we're going to track the inputs and the outputs. So what happens is light and water enter the chloroplast. So the light enters by getting absorbed through the leaf and then into the cell. The water would enter initially by coming into the plant through osmosis via its roots. The water gets transported throughout the plant by a vessel called the xylem, and it would get delivered to these cells in the leaves, and once again through osmosis it would make its way into the plant cells and then make its way into the chloroplast, where it's going to get used up. So light and water enter the chloroplast, and specifically they enter the thylakoid membranes. The light and the water interact with one another, and essentially the light causes the water molecule to be split apart, and if a water molecule is split apart, H2O, it's split into 2 hydrogen ions and 1 oxygen atom. Now, That happens to two water molecules. Well, it happens to all of the water molecules, but the light splits the water. The oxygen will fuse with another oxygen and it will leave the chloroplast, and it'll travel into the plant cell, and it could leave the plant cell and travel out of the plant into the atmosphere, or it could get used by the plant cell in cellular respiration. So we've used our water. Water's gone. It's now being converted into hydrogen ions and oxygen atoms. The oxygen atoms have left, so they're gone now too. With the hydrogen atoms, they're picked up by a coenzyme called NADP+, and once that coenzyme, which we call an acceptor molecule, once it's loaded with the hydrogen ions, it becomes NADPH. And it's actually going to transfer those hydrogen ions over to the next stage of photosynthesis, called the light independent stage. Now, whilst that light has entered and been absorbed by the chlorophyll, along with causing water to be split, it's also caused a series of reactions to occur that has produced ATP. So coming into the light dependent stage, we've had ADP plus the inorganic phosphate, and using sunlight, that's provided us with energy to bind the phosphate to ADP to regenerate that into ATP. And like the NADPH, ATP is going to go over to the second stage of photosynthesis and get used up. So light is absorbed by chlorophyll and splits water to produce electrons, hydrogen and oxygen. Absorption of light also causes ATP to be formed. Oxygen is released to the atmosphere as a byproduct, or it's used in cellular respiration by the plant. The ATP and the NADPH is moved to the light independent reactions to be used there. Here's a consolidation task for you to try and fill out. I won't draw a diagram on there for you, but I'll go through the answers to your table. So once again, maybe pause it now and have an attempt at doing it yourself. So if we're looking at the inputs and the outputs, in a question like this that wants you to list multiple inputs and multiple outputs, it's asking for the inputs and outputs shown by the chemical equation, but it's also going to want you to list the other like minor inputs and outputs that are occurring as well. So we're just talking about the light dependent stage. So if we start with our input from our chemical equation, our main input there is going to be water, H2O. I might just go through the answers instead of drawing them. So if our input here is going to be H2O, our main output from the equation is going to be oxygen. Some of our other inputs and outputs, an input would be ADP plus PI, and its output would be ATP. Another input would be NADP plus, our coenzyme, and its output, once it's loaded with hydrogen, would be NADPH. So let's have a look at the second stage of photosynthesis now, which we refer to as the light independent stage, also known as the Calvin cycle. And like I said before, to get a good understanding of this, you really need to watch that other video called Photosynthesis, which will map it out really nicely for you. But here's a quick overview. So we've got our light independent stage shown there. We've already talked about the light dependent stage. The light independent stage is where we take in carbon dioxide, and this stage occurs in the stroma of the chloroplast. So in the stroma we have this region where a series of reactions takes place that we call the Calvin cycle. So the Calvin cycle is essentially a biochemical pathway composed of lots of different enzymes that catalyse lots of different reactions. We don't need to go into detail with that. What we do need to know is that entering the Calvin cycle, we have carbon dioxide. And that carbon dioxide is taken into the plant out of the atmosphere, or it could have been produced by the plant during cellular respiration as well. We use the Calvin cycle to convert carbon dioxide into glucose, which is the sugar we're going to store in the plant and that the plant's going to use for energy. So the Calvin cycle converts carbon dioxide into glucose, and a good way of saying that is carbon dioxide is reduced into glucose, because reduced is the chemical term we use when we're building something up. Now, as you can see, glucose is a molecule which has carbon, hydrogen and oxygen. Specifically, it has 6 carbons, 12 hydrogens and 6 oxygens, C6H12O6. The carbons in glucose are going to come from the carbon dioxide that enters the Calvin cycle. The oxygen in glucose is also going to come from the carbon dioxide that enters the Calvin cycle. However, as you can see, we also need to incorporate 12 hydrogen molecules to actually formulate that glucose. So back in stage 1, if you remember, when we split water, we produced hydrogen atoms and we loaded them onto the coenzyme NADP+, and I said NADP+, which then becomes NADPH, transfers them to the Calvin cycle, and that is our source of hydrogen. So by using the carbon dioxide that enters the Calvin cycle, along with the hydrogen delivered to the Calvin cycle via NADPH, we are able to combine those to produce glucose. Now that's all well and good, but it will not occur if we don't have energy in the form of ATP to drive all of those chemical reactions within the Calvin cycle. And that's why during the first stage, the light dependent stage, when we use the sunlight to produce ATP, that ATP is going to get used up in the Calvin cycle to fuel or drive that process of converting carbon dioxide into glucose. So we could say carbon dioxide enters the Calvin cycle, ATP and loaded acceptor molecules NADPH from stage 1 drive the Calvin cycle to convert carbon dioxide into glucose. The glucose is then stored in the plant. ATP and NADPH are cyclic molecules, so once they've done their job, their unloaded forms will travel back to the light dependent stage to do it all again. And even though we call this the light independent stage, hopefully you can work out it still would not occur in the dark, because the Calvin cycle will not work without the addition of the hydrogen and the addition of the energy in the form of ATP, and we can't get those two things unless the light dependent stage is working, and that stage won't work unless there's sunlight. Here's another consolidation task for you to fill out. So in terms of inputs of the light independent stage, from our chemical equation, our main input is carbon dioxide, and also from the chemical equation, our main output is glucose. Our other inputs and outputs, an input would be ATP, and the corresponding output would be ADP plus PI, and an input would be NADPH, and a corresponding output would be NADP plus. This is a really detailed diagram showing the Calvin cycle, and it's definitely 100% not something you need to know or be able to talk about. What I'll point out though is what it shows us is that the Calvin cycle is composed of a series of chemical reactions, just like I said before. And at each reaction, a reactant is getting converted into a different product. That product is becoming the reactant of another reaction, and so on and so forth. What this diagram does show us is often we talk about the Calvin cycle as carbon fixation, where we say the carbon dioxide molecules are sort of fixed into the Calvin cycle. And I just want to point out two things. One is that we have this molecule called ribulose biphosphate, RUBP, and this molecule spends its whole life in the Calvin cycle essentially and just gets converted into different forms. What we do in the Calvin cycle is carbon dioxides are added to RUBP, and that's how they enter the Calvin cycle. But to add them onto RUBP, we need an enzyme to catalyze that chemical reaction, and that enzyme is called ribisco. You might see that used in different questions to sort of create a context of a question. So what I want you to think of is ribisco is the enzyme in the Calvin cycle that is responsible for carbon fixation, for incorporating carbon dioxide into the Calvin cycle. And down the bottom here talks about a sugar called G3P. So what actually happens is the Calvin cycle produces these 3 carbon sugars called G3P. 2 of those combine to form a 6 carbon sugar, which we refer to as glucose. Photosynthesis is a process where the rate in which it takes place, so the rate in which it's producing oxygen and glucose, can vary. And it varies because the process is dependent on different things. So the process is dependent on light. It's also somewhat dependent on temperature, and it's obviously dependent on providing carbon dioxide. So we would say these are 3 factors that can affect and influence the rate of photosynthesis. Term down here, a limiting factor. A limiting factor is a factor that is restricting the rate of photosynthesis if it's in short supply. So if there's not enough carbon dioxide, that's a limiting factor, even if there's plenty of light and an appropriate temperature. Likewise, if there is lots of carbon dioxide and appropriate temperature, but a low light intensity, we would say the light intensity is a limiting factor because it's the thing that's limiting the rate of photosynthesis because it's in a short supply. Any of those 3 things can become a limiting factor depending on the situation. So with light intensity, as the intensity of light increases, the rate of photosynthesis will increase linearly to that increase. So the higher the light intensity, the higher the rate of photosynthesis. Reason why is if there's low levels of light, then the light dependent stage of photosynthesis will take place slowly. So it'll be producing ATP and NADPH relatively slowly. So it'll be a slower supply of those things to the Calvin cycle. Therefore, it will be a slower rate in which it's able to make glucose. On the other end of the spectrum, if there's high light intensities, you're producing ATP and NADPH quite quickly. They're getting delivered to the Calvin cycle quickly, and the process of photosynthesis is taking place quickly. You can see by this graph, I told you that there's a linear increase, which there is, but you could get to a point where there's a plateau. And what the graph's showing us is that despite continual increases in light intensity, the rate of photosynthesis has reached its peak, and it's not going to increase any further. What accounts for the plateau? What accounts for the plateau is there's a different limiting factor acting on photosynthesis at that time that's not light intensity. So carbon dioxide concentration could be the limiting factor, or temperature could be the limiting factor. And like I said before, if you have a limited supply of carbon dioxide, it doesn't matter how much light intensity you add, the photosynthesis will only occur at the rate in which the carbon dioxide supply is allowing it to. In terms of the effect of carbon dioxide concentration, it's exactly the same trend and relationship as temperature. The increase in the rate of photosynthesis is linear to the increase of carbon dioxide concentration. The more carbon dioxide there is, the faster the rate of photosynthesis. That's because the faster the rate that we're supplying the carbon dioxide to the Calvin cycle. However, once again, as you can see, we can get to a point where the graph will plateau. Once again, what accounts for that plateau? Well, something besides carbon dioxide concentration has now become the limiting factor of the rate of photosynthesis. So maybe now we don't have a high enough light intensity, or we don't have an appropriate temperature. In terms of the effects of temperature on the rate of photosynthesis, you'll notice I've been pretty careful in the last two slides and I've said an appropriate temperature. That's because it is all related to having an appropriate temperature. So what you will see is the rate of photosynthesis will increase with an increase in temperature up to a point, and that's the optimum temperature for photosynthesis to occur at. After that point, the rate of photosynthesis will decrease, and it'll decrease pretty dramatically. It's the same trend as temperature for enzymes, and it's the same trend because it's the exact same thing causing the change in the rate of photosynthesis. Photosynthesis is a process driven by enzymes. We know that enzyme activity increases with an increase in temperature until that enzyme's working at its optimum temperature. So that's why photosynthesis increases as temperature increases, because the enzymes are working faster, so the process is taking place quicker. Once we get to the optimum temperature, photosynthesis is occurring at its fastest rate. If temperature continues to increase, the enzymes responsible for photosynthesis will denature, so therefore the rate of photosynthesis will drop, and it'll eventually drop back down to zero, because the process won't occur at all if all of the enzymes are denatured.